Hello and welcome to today's Odyssey. In this episode, we will discuss one of the most cutting-edge and controversial topics in neuroscience: organoid brains. What are they? How are they made? What can they do? And what ethical issues do they raise? Let's find out. Organoid brains, or brain organoids, are miniature 3D structures that resemble parts of the human brain. They are created by culturing pluripotent stem cells, which are cells that can turn into any type of cell in the body in a special device that mimics the conditions of the developing brain. Over a course of months, the stem cells differentiate into various types of neurons and other brain cells, and self-organize into complex networks that resemble regions of the cortex, hippocampus, retina, and other brain areas. Organoid brains are not exact replicas of the human brain, nor do they have the same size, shape, or function. They lack many features of the mature brain, such as blood vessels, immune cells, glial cells, and connections between different regions. They also do not have sensory inputs or outputs, meaning they cannot perceive or interact with the environment. However, they do have some remarkable abilities that make them valuable for research. Organoid brains can generate electrical activity similar to brain waves that indicate some level of communication and coordination among neurons. They can also respond to stimuli, such as light or chemicals, and show signs of learning and memory. Some organoid brains even exhibit spontaneous movements, such as twitching or contracting. These behaviors suggest that organoid brains have some degree of self-organization and plasticity, which are essential for brain development and function. Plasticity is the ability of the brain to change and adapt in response to experience or injury. Organoid brains are useful for studying how the human brain develops and works, especially in comparison to animal models, which have limitations in terms of genetic similarity and ethical acceptability. Organoid brains can also be used to model human neurological diseases, such as Alzheimer's, autism, schizophrenia, and epilepsy. By using stem cells from patients with these conditions, researchers can create organoid brains that carry the same genetic mutations and observe how they affect the structure and function of the brain cells. This can help to understand the causes and mechanisms of these diseases, as well as to test potential treatments. However, organoid brains also raise some ethical questions and challenges. One of them is whether organoid brains have any form of consciousness or sentience. Although organoid brains are far from being conscious in the human sense, they may have some rudimentary awareness or experience of their own existence. This could pose a moral dilemma for researchers who manipulate or destroy them for scientific purposes. Another issue is how to regulate the use and disposal of organoid brains. Currently, there are no clear guidelines or laws that govern the creation and handling of organoid brains. This could lead to misuse or abuse of these biological materials by unscrupulous actors. Organoid brains are a remarkable achievement of science and technology that offer new possibilities for understanding and treating human brain disorders. However, they also pose new challenges for ethics and society that need to be addressed carefully and responsibly. As we continue to explore the mysteries of the brain with organoid brains, we should also consider the implications and consequences of our actions. Thank you for watching today's Odyssey. If you enjoyed this video, please like and subscribe for more, and don't forget to leave your comments and questions below. See you next time.